All right. All right. I have a couple of more slides. I think after all these formulas, I owe you a little bit to tell you why actually we're doing this, and then I'm also gonna try to tell you what actually our current state of research and our current state of understanding is in this field. So you had Johannes' lecture last Friday about OCT. So you've seen a couple of OCT images already, and he did mention the attenuation coefficient in there. So I want to elaborate a little bit on that attenuation coefficient. Using this example. So here you see a drawing of the urinary tract with the kidney. The ureter is the channel that guides the urine to the bladder, and there it is collected and eventually disposed of. Urethral cancer occurs in the urethra as well. Unfortunately, of course, and the result of that is that that urethra is blocked and kidney failure can occur. So it is important to diagnose that accurately and to find it earlier on, so that an intervention can take place. It's a very small organ, so it is hard to see on the conventional techniques. You cannot really see it on MRI or CT because that resolution is not good enough. Also not on ultrasound, so it is really hard to diagnose these tumors actually. One of the things that may be able to do that is OCT, and then from the inside. So let's assume that there is a lesion over there. That's the green dot there. And the idea is to go there with a probe, make a circumspherical OCT image. So the probe is inside the urethra and looks around, collects scattered light all around to make up an to make an OCT scan of a small segment, and then hopefully that segment contains the lesion that can then be found through image processing. So this way you can visualize the lesion, you can estimate how large it is, how etc., and how the surrounding tissue looks like, if that is affected or inflammatory or anything else. So you can hopefully that's the goal, to use that for planning an intervention. So on the left-hand side, you see two stills from such a rendering. So so the probe is again inside this lumen, and then collects reflected light all around. So you can plot it then on this circular plot. And you can see two lesions here. So here is the as you can see, as kind of an outgrowth. You can see the tumor, very small. And one of the things that the doctors really would like to know is whether this tumor is growing in or not. So there is this small membrane that separates the outside tissue from deeper tissue layers, the so-called basal membrane. And once a tumor breaks through, so if it grows through the basal membrane, then the risk of spreading becomes very high. If it is not through the basal membrane yet, then usually an intervention can be done, and then the outcome is very good. So one of the holy grails, or one of the things that we would really like to see on these images, is whether or not the basal membrane is still intact. So that is the top image, and then on the lower image you have a more blurred image. It is not really possible to see any structures anymore in it, so that and that is usually associated with a tumor that is very aggressive. What the exact physical or the exact biological mechanism is, that I do not know, but usually it is something along the lines that if the tumor is very aggressive, then it really does not. Then it puts all its energy in growing and not so much in making sure that the environment is cleaned up. So it's usually really sloppy, high metabolism cells that appear in these images as yeah, just layerless blobs. Now, what so the idea is so both actually are features that that can be that can be seen in the image, and there's also histology to back that interpretation up. So so in this case, it was possible to take biopsies after imaging and then find the exact same spots again where the image was taken. And then in the histology, it was possible to show that the basal membrane in this case was actually still intact. So that was for this lesion was good news. And also the disappearance of tissue structure can also be seen in these pathology images. Okay, so staging is then the stage of a tumor is then the invasiveness. So a higher stage means that it has grown deeper, and the highest stage means that it has actually passed this basal membrane. But that is something that can be that can basically be elucidated from the image itself. But then the grade, the aggressiveness, you can see some disappearance of structure from the image itself. So you can see that something is wrong. But the amount of aggressiveness, the grade number, so to say, or the grade scale is hard to see on the image. And then our hypothesis has been for a couple of years now that we can do that by measuring the optical properties of such a region. And see if we can find differences between the optical properties in order to do this grading. That turns out to be feasible for this tissue type. At least it was possible to measure these attenuation coefficients from OCT images. So what you're basically doing is you measure how fast the signal decays as you look deeper. A faster decay, just like in Lambert-Beer's law, a faster decay means a higher attenuation coefficient, and a slower decay, a lower attenuation coefficient. And in this case, it was found that a high-grade tumor, so the more aggressive tumors, also had a higher attenuation coefficient, and that the lower-grade tumors had a lower-grade attenuation coefficient.
I have a question about the graph.
Yes.
In the left image, you said that in the lower image of the two, that the structure is missing and that therefore there is a tumor. But to be honest, it didn't occur to me that the structure was missing there, because if I'm looking to the not so zoomed in image, then it seems to me like this part of the wall is more or less the same as any other part of the wall on a circle. So how could I see that here the structure is missing? What would I normally see?
So what you would normally see is a little bit more. It's a little bit hard to see because this in the top image, but it's a little bit hard to see because the tumor is there. Is more like a layered structure. So you see a kind of like individual cylindrical layers stacked on top of each other, and that is a little bit missing in the lower image, especially at the location of the arrow. So you do see one little bit darker top layer, but after that it is less visible. And then again, I'm not a doctor,
Okay.
so I'm not entirely sure if that
No,
that
okay.
explanation is totally true. But that is usually what's being looked at.
Okay.
So, but back to the grade and to the attenuation coefficient. So, like I mentioned, we've no, one second before, one thing before. So an interesting question is why is this happening? So why does a higher grade, a higher aggressiveness of the tumor have a higher attenuation coefficient, and a lower grade have a lower attenuation coefficient? We've measured this not only at these urethral tumors, but also at other tissue types. And sometimes it's like this. So the more aggressive tumor has a higher attenuation coefficient, but sometimes it's also just the other way around. So then the difference can be found, but the less aggressive tumors have a higher attenuation coefficient, and for some tissues there is no difference at all, not a distinguishable difference. So the question is, basically what's the underlying mechanism here, and that's what we try to understand in our research. So the idea or the concept is more or less clear. So if you have normal cells that are well behaved, that have regular shapes and regular organelles in there, they will give some kind of scattering signal. And then when cancer occurs, it's known just from histology that changes occur. So multiple nuclei can occur in the cell, for example, or more mitochondria because the cells are so metabolically active, and their shape changes. So this will cause. We can expect that this will lead to a change in scattering properties, but how exactly is not so well understood. But asks, does the attenuation coefficient also depend on the radiation doses? No, in principle. So you mean for imaging, or do you mean for if you would do a treatment? If you would do a treatment for imaging? No, it is the relative. So it is really like Beer's law. So you measure I is I0 exponential minus mu. In this case, attenuation coefficient minus mu t times distance. So it is really this mu that is in the attenuation that is measured here. So it is a relative measure. Yeah. So what you then basically try to do is interpret. Or we expect that the change in physiology of the tumor will lead to something that is quantifiable from an image. So like a quantifiable change in optical properties such as absorption or scattering coefficient or anisotropy, could be any of those. But the real challenge is understanding how that mechanism works, and then at some point switch this around. So if we measure certain optical properties, can we then say something reliably about the physiology? I'm going to skip through this. Yeah. So we did try it in a couple of tissue types, tumor types. Actually, the list is a little bit longer today, so I have to update this graph at some point. But sometimes the point of this list basically is that sometimes we could distinguish between normal and diseased tissue, and sometimes we simply could not. So basically our physical scientific approach to solve this problem is I like to call it kind of a stepping stone approach, where we approach the problem in steps and from two sides. So the gold standard is and remains the pathology. So but taking away tissue and looking at it under the microscope is not always possible, but it remains the gold standard. So on that side, the research that we do is we have all this pathology, we digitalize it, and then we try to extract as much as possible information from these digitized slides. So as much information as possible that we think that might be causing changes in scattering properties. So we try to segment nuclei. We try to measure cell sizes or distribution of cell sizes, etc. And having the assessment of the pathologist, we can then try to relate that. So that is on the let's say on the reference side, and then on the optical side, we start out with the scattering properties that we quantify from in this case the OCT images, so the scattering coefficient, the absorption coefficient, and then to interpret that back into some kind of pathology, we need models for the light scattering, and that is what we have been doing in this course now for the last couple of sessions. So one of the possible models is that you model tissue as a collection of spheres, as this discrete random medium, and that no, we came pretty far with that. We could calculate the scattering properties of a single particle, and with the structure factor and pair correlation function, we could calculate the scattering properties of a collection of particles. So as far as the model goes, we could do that, and also because we have the formulas and we have the theory behind it, we can also interpret back that if we measure some kind of some set of optical properties, we can for example, interpret that in, let's say, average particle size or average distance between particles, something like that. So, so in a way that works. Where it breaks down, of course, is when tissue is not a collection of particles, which is basically always. So there we need a more sophisticated model, and that actually was that model is actually the last topic of the class of today, is modeling tissue not as a discrete random medium, but as a continuous random medium. So we don't have particles now; we just have one big soup of refractive index fluctuations that will scatter light. Of the question. Yeah, of course, yeah. So yes, could this digital pathology be used in combination with machine learning and pattern recognition? Yes, of course, and that's of course we do that as well separately. Yeah, definitely. That is a big topic of research these days. Yeah, there's a lot of interesting things to say about that, but I'll not get carried away on that for now. So this model that we would like to use, but which turns out to be very difficult. So really this is now about the point where we are in the science, trying to model this continuous random media and trying to understand actually what we're modeling. So let me talk you through a couple of things of that. So we have no distinct particles anymore, but a continuous spatial variation in the refractive index. So this M, so relative refractive index or refractive index squared, is proportional to the density. So that gives us then the link, a little bit of a link to the physiology, that if tissue is more dense, it's one of the changes that can happen during disease progression, then we'll expect a change in refractive index. To make things a little bit manageable, we assume that the distribution of M, so this or M squared, can be characterized by two values only. So you don't need to have the whole distribution. If you know the mean and the variance, then that should be enough to calculate the optical properties. But instead of discrete particles, we have now defined it as a random, the refractive index now as a random quantity. So we cannot simply calculate M squared because that then the answer would depend very much on which random value we were actually probing. So instead of calculating M squared, we now have to calculate correlations of the refractive index. So a spatial correlation basically means that you're trying to investigate on what scale is my refractive index fluctuating. So if that correlation is very long, then you have a very slow fluctuation of refractive index, and you can expect that there will not be a lot of scattering. If the fluctuation is very fast on short length scales, then we can expect higher scattering signals from that. Now going back to our famous integral that contained M squared for if we calculated it for a particle. So this is then the Rayleigh Gans. Again, the Rayleigh Gans integral for a particle. So instead of calculating now this one integral or this one factor, we also calculate an average for that. We have to calculate an average for that, because now if M is random, then this S will also be random. So we need to calculate the average. So instead of M squared here within the integral, now comes this correlation function, this integral correlation function. And otherwise, the formalism stays the same. So it is still a Fourier transform, but now of the correlation function, and there is still this angular dependence that corresponded to the dipole scattering pattern. So we're still considering this sort of random fluctuation, this whole soup of refractive index variations to be made up of individual dipole scatterers now. All right, so if we then look at the optical properties, then much of it just stays the same as it was. So the scattering coefficient is still the volume average of the total scattering cross-section. The total scattering cross-section is found by by integrating this differential scattering cross section. So basically, this contains both the scattering cross section and the phase function. By integrating that over all angles, and then where it is different now is in the expressions for this differential scattering cross section, for this sigma theta phi. So in the previous applications, we calculated that using Rayleigh theory or Rayleigh Gans or Mie scattering, but now that formalism for this continuous radial medium becomes a little bit different. So if you look at the factors here, we have a lot of since we consider the sample being built up of dipoles, we have a lot of the characteristics of dipole scattering here. So this is the dipole scattering pattern that is in there. This is the dependence on the wave number to the fourth power. That is also characteristic of it. And then here we have the function that needs to tie this all together. So what was basically what was the form factor in the Rayleigh Gans theory now becomes this power spectral density or the Fourier transform of the refractive index correlation function. So the B is the correlation function of the refractive index. This takes the Fourier transform, gives us this function phi of Q. Q is the scattering factor 2 pi over 2k times sine half theta, and then we can express that as a function of theta and phi as well. So we have the dipole scattering pattern that depends on theta and phi, and we have this correlation function that also has theta and phi components. Yeah, so again, B is then the correlation function of the refractive index, and Q is the scattering factor. And this is where it really gets complicated, because so far this is a little bit an exercise of replacing discrete quantities with random quantities and replacing straightforward integrals with correlations. But how to model this correlation function? And then if you go in the literature, then there is one very popular choice, so-called Whittle-Mattern correlation function. Actually, it's not a correlation function, but a so-called family of correlation function, because it has a couple of parameters which you can use to tune the appearance of that correlation function. So there is 3 parameters that describe this correlation function. One is the variance of the refractive index distribution. So if you remember on the in the first slide of this topic, we assumed that we could characterize this distribution with its mean and its variance. Now the variance is here. Then there is a function, a parameter beta that basically governs how the shape of this function looks like. So if beta runs is no, it's not necessarily an integer. I think it's if it is a low value, so close to 0, then you have a kind of an exponential function, I think. And if it goes to higher values, then it becomes more Gaussian shaped. But the specifics are not that important now. And then we have the correlation function, the correlation length. So that tells us on a spatial scale how fast this refractive index fluctuates. So again, a long correlation length means that it is a slow fluctuation, and a very short correlation length means a strong fluctuation. And then there is a few functions. So there's a gamma function in there. There is I don't even remember what a k function is. But basically what this does is that this throws us off of our familiar way of approaching scattering. So we were used to calculating scattering using the refractive index of the particle and of the medium, using the size of the particle and the volume fraction of the particle. If we had those things, we could calculate everything. And now all of a sudden we have to use the variance of the distribution. We have to use the shape parameter, and we have to use the correlation length. So that makes it a little bit challenging to tie all these knots together. The good thing about doing this, and that's why we still think it's promising, is that whatever you can get statistically from pathology is something that you can put in this equation. What I mean with that is that if you have a pathology image, you can do just an image cross correlation and see at what length scale the image changes. So if that is on a long scale, so that means that that the image is really self-similar over a very long distance, then we can estimate that this correlation length that goes in here will also be long. If we see a lot of intensity fluctuations in this image, then we can also expect that the variance of the index distribution will be very high. So we can assess a little bit of some of these parameters. We can probably assess directly from pathology, so that is good news. But yeah, the drawback is my complaint earlier that we have a completely new set of parameters that are not so easy to understand. This is the most potent way to actually measure this refractive index distribution. We can use phase microscopy. Phase microscope gives you a direct map of the refractive index of a thin sample. So from an image like this, from a phase microscopy image like this, we could directly calculate this distribution and its correlation function. So it is still a very promising route. All right. This leads up to assignment 2 that I will hand out on Friday, and it will be about this paper. So reading that paper, or at least reading the first half of that paper, is going to be homework for Friday. And then on Friday, I want to discuss the actual assignment. So I want to start on Friday by discussing this actual assignment. What I really like about this paper is that it tries to that it acknowledges that if you go through this whole new formalism, that you have some work to do to interpret it. And the really good thing about this paper is that they take one sample, the sample of spheres, a collection of spheres, and try to model it using both formalisms. So once as a discrete random medium, like we have done, or let's say almost like we have done, and using this new formalism. I think, I hope that with what we have discussed today, you have enough background to go through this paper. So that is basically the first thing that I want to see. I also know that I have received the assignment 1 from some of you, but not from all of you, and we had set the due date for that for the next class. So I'm going to split this a little bit in two. So the assignment will follow on Friday, and reading this paper is going to be homework. I'll send an announcement later today with some more details.
Will the deadline of assignment 2 also be the next lecture on Friday?
Yep. So 1 week later, and then the deadline for the third assignment will be before the oral exam.
Okay.
Yep. Okay, that's it for today.
All planned in for the oral exam in 3 hours on Friday morning?
That seems virtually impossible.
Yeah.
I think so. We have to. No, no. We have to plan that in some way. So some of you will, but I guess for some of us, we have to make different arrangements.
I believe it's not on Friday in the morning because that was the career day, and I thought that it would be shifted to another date, right?
I don't know actually. So
okay.
I'll look that up, and let's strategize about that on Friday, on next Friday as well.
All right.
Okay.
Yep. Okay. That's it then.
All right.
Have a
See you
nice
Friday.
Okay. Bye.
evening and afternoon.